introductory part, then types of tannins, then chemical test, and a few important drugs from tannin category. So first of all, yeah, what do you mean by tannins? So these are also secondary metabolite of plant. They are also secondary metabolite of plant like your alkaloids and glycosides, and they are also most widely occurring in the higher plants. These secondary metabolites occurs in the form of cell sap. Cell sap means the space present. These are also secondary metabolites of one plant that are occurs in the higher plant mostly. The higher plant होते हैं उनमें आएगा that occurs in the form of cell sap. Now these tannins have one unique property. They can precipitate your proteins. These tannins will have a specific property that they can precipitate your protein. Okay. They can combine with the protein and precipitate them. So they are the protein precipitants. That's why they are sometimes used as an antiseptic to block the hemorrhage, small hemorrhage. So tannins are again these are nothing but the secondary metabolites of the plant. They particularly occurs in the higher plant and they are found in the form of cell sap or distinct. They are astringent substances that is nothing but they have capacity to combine with the protein and precipitate. They are nothing but the organic compound, non-nitrogenous. Remember, organic non-nitrogenous derivative of polyhydroxybenzoic acid. So alkaloids are nothing but they will have nitrogen in their structure. Some glycosides will also have nitrogen in their structure. These tannins are not nitrogen containing compound. These are organic non-nitrogenous and they are derived from the benzoic acid. They are the derivatives of polyhydroxybenzoic acid. They are used in the medicine for different purpose. Like they will act as a mild antiseptic due to their protein precipitation property. Then they are also used in the treatment of diarrhea. They will cause constipation and they are used in treatment of diarrhea. Then again they are also used to small hemorrhages, not a hemorrhage from small artery or from capillaries you can say. So to check the small hemorrhage or to check means to stop or to choke to stop the hemorrhage from small. They are extensively used in the leather industry for the tanning purpose. So these tannins they are extensively used in the leather industry. So to tan the leather. Chemically, they are a mixture of polyphenol, okay, polyphenol generally ortho or diortho or tetrahydroxy groups on a phenyl ring. So phenyl ring will be there 2, 3, 4 hydroxyl group. Simplest kind of tannin is catechol nucleus. So catechol. Chemical properties of tannins. They will form a colloidal solution with water. They are non-crystalline substances. Acidic reaction due to presence of polyphenol groups. They are soluble in alcohol, glycerin, dilute acid, but they are practically insoluble in the organic solvent except acetone. So your alkaloids are soluble in the organic solvent, but these tannins are not soluble in the organic solvent. They are soluble in the alcohol, glycerin, and dilute. They will form colloidal solution with the water. These are the properties of tannins. Specific taste. This part is important for your specific taste for. So as they have protein precipitation power, so this gelatin is not about the type of protein. So it is utilized to taste the presence of your tannins. So they can precipitate this protein that is known as a gelatin. So gelatin precipitate. So tannins will precipitate and alkaloids. Tannic acid particularly they can precipitate alkaloids. That's why this tannic acid is a part of it. Then precipitation test again. Tannins are precipitated by copper salt. Tannins are precipitated by copper salt, lead and tin. So salts of copper, lead and tin they can precipitate your tannin. Again tannins are also precipitated by strong potassium dichromate solution and chromic acid solution. So this is the precipitation test. No one is going to ask you this test. We are going to ask you regarding this test. Color reactions with iron salt. Color reactions of tannins with iron salt. So this ferric chloride will give you bluish black or greenish brownish green color with this ferric chloride will give this green bluish black or brownish green color. Then with potassium ferrocyanide and ammonia, they will give you deep red color. Deep red color is done with the potassium ferrocyanide and potassium ferrocyanide. These are the color reaction. This is the very specific test for identifying gold bitter skin test. Gold bitter skin test. So here a gold bitter skin that is a untanned skin from animal. इट इज नोन एज गोल्ड बेटर स्किन. किसको गोल्ड बेटर स्किन क्यों बोलते हैं वो छोड़ो. गोल्ड बेटर स्किन मतलब एनिमल स्किन, अनटैंड एनिमल स्किन. सो अनटैंड एनिमल स्किन डिफरेंट फ्रॉम मेम्ब्रेन इंटेस्टाइन ऑफ ऑक्स के इंटेस्टाइन से जो स्किन मिलता है उसको बोलते हैं गोल्ड बेटर स्किन. You have to wash that skin with the distilled water and hydrochloric acid and rinse with the distilled water tannin solution, or place tannin solution for wash with distilled water, add ferrous sulfate solution. You will get a brown or black color on the skin. That is due to presence of, that is nothing but a gold beater skin test. It is a specific test for identity. Next is the pinazone test. So tannins are precipitated by 2% solution of pinazone. Tannin solution should be prepared with the sodium acid phosphate. First tannin solution you have to prepare with the sodium acid phosphate, and then you have to add pinazone solution. Tannins are precipitated by 2% solution of pinazone. You will get precipitated. Then next test is the vanillin hydrochloride test. Vanillin is Cl test. So vanillin 1 gram vanillin plus 10 ml alcohol, 10 ml concentrated HCl. You will get a red color. Tannins are present, particularly hydrolyzable type of tannins are present. Then only this test is given. Vanillin hydrochloride acid test. Due to there is a formation of chloroglycerol. So what happens? What happens in this test? What happens in this vanillin hydrochloride acid test? So initially, because of this acid, these tannins are hydrolyzed and they are converted into they are degraded. Okay? And this chloroglycerol is a degradation product. Okay, hydrolysis product of tannins. And then this chloroglycerol react with the vanillin to give red color. Next specific test for tannin, matchstick test. Matchstick test. This test is again important. Remember, vanillin hydrochloride test is important. Matchstick test is important. Gold beater skin test is important. So now what happens in this matchstick test? So this test, the matchstick जो आप काडीपेटी आहे, काडीची पेटी जी आहे त्याच्या स्टिक असते. That matchsticks you have to dip in the aqueous extract of the tannin solution. You have to place or plant near to the burner. You have to moisten that stick with the HCl. And you have to place near to the burner. Then this stick will burn with the red color, burn with the red fume. That indicates the presence of chloroglycerol, which is a hydrolysis protocol. That is a matchstick test. So again, these tests are very important. You have to remember these tests. So gelatin test, protein precipitation with the different salt like copper, potassium dichromate solution, precipitation with potassium dichromate solution or formic acid. Color reaction with the perichloride, bluish black or greenish brownish green. With the potassium ferrocyanide, and ammonia, you will get a deep red color. Then gold beater skin, that is a skin of intestine of ox. Then with it will give brown or black color. So that's why these tannins are used for tanning the skin in the leather industry. Pinazone test, then vanillin hydrochloride, hydrochloric acid test. Then matchstick test. These are the important tests for identification of your tannins. ये टेस्ट याद रखो. मैस्टिक टेस्ट, गोल्ड बेटर टेस्ट. देखो इसमें एक ऐसा है, आपको मिक्स क्वेश्चन भी डालते करते हो. सो एक इसमें मतलब कोई इसमें देगा डिफरेंट सेकेंडरी मेटाबोलाइट, अल्कलॉइड, ग्लाइकोसाइड, टैनिन्स, फ्लेवोनॉइड्स. ऐसा एक कॉलम में देखिए आपको ये 4 अल्कलॉइड्स, ग्लाइकोसाइड, टैनिन्स एंड फ्लेवोनॉइड्स. और दूसरे कॉलम में देखिए उनके स्पेसिफिक टेस्ट. जैसे होता है अल्कलॉइड के लिए हैगर्स रिएजेंट, मेयर्स डायग्राम ड्रॉप, उसमें का कोई भी देगा. देन ग्लाइकोसाइड के लिए होता है डिफरेंट टेस्ट, बालजेट टेस्ट, लीगल टेस्ट, टेलर किलनी टेस्ट, देन ब्रॉन ट्रैकर टेस्ट. ठीक है, तो इसमें का कोई भी टेस्ट देगा. और तीसरे कॉलम और देगा आपको कि मैच स्टिक स्टेज टेस्ट, और आपको देगा सिनोडल टेस्ट. एंड क्वेश्चन विल बी लाइक दिस, मैच द फॉलोइंग सेकेंडरी अल्कोलाइड एंड देयर आइडेंटिफिकेशन टेस्ट. ये सभी क्वेश्चन आ सकता है. इजी होता है वो, सिर्फ आपको कंफ्यूज करने के लिए होता है. आप इजीली आप उसको मैच द फॉलोइंग कर सकते हैं. आपको अगर 2 या 3 मैच भी मालूम है तो एक करेक्ट आंसर मिल जाता है. Tannins are nothing but the classified into different type. Tannins are classified into hydrolyzable tannins and condensed tannins. There are two class of tannins. One is a hydrolyzable, second is a condensed tannins. Okay. So what is the chemical structure for tannins? All the tannins or the plant containing tannins, they consist of they will have a derivative of these structures. Gallotannins, elagitannins, complex tannins, and condensed. These they are nothing but the polyhydroxy phenolic. So this is known as the flavon. This structure is known as the flavon. This structure, for example, they are known as a this structure is known as ellagic acid. Ellagic acid, or this structure is also ellagic acid. So these are not derivative of your. They are derivative of this structure only. So these are the structural. So basic structure of tannins will look like this. Then again, individual structure. You can see gallic acid, then ellagic acid, phytocatechol, catechin. So gallic acid will look like this. 3 hydroxy group with benzoic acid को 3 additional hydroxy group डालो, that is gallic acid. Ellagic acid will look like this. This is pyrocatechol, pyrocatechol, or this is catechin, catechin, epicatechin. These are the constituent. This is catechin. These structure are nobody is going to ask you this structure. Just for your information, I will see the individual classification of your tannins. This part is important. They can ask question on this. So you have to remember the classification. First of all, classification of tannins. So they are classified into these classes. First one is a hydrolyzable tannin. As the name indicates, hydrolyzable means you can carry out the hydrolysis. They can be hydrolyzed by different acid and enzyme. So they can be hydrolyzed by acid or enzyme quickly. So and gallic acid, then ellagic acid are the hydrolysis product. So after hydrolyzable tannin, you will get a gallic acid and ellagic acid. And upon dry distillation, gallic acid and other component gets converted into pyrogalal. And this pyrogalal with ferric chloride give blue color. Examples of your hydrolyzable tannin. So gallotannin present in the which is also present in rhubarb, clove, chestnut, elagitannin from oak, and myrobalan. The color of sometimes the color of your pomegranate bark is due to the tannin. So these are the hydrolyzable tannins. Condensed tannins क्या होते हैं? So obviously they are not hydrolyzable. So they are not hydrolyzable. Probotannins, proanthocyanidins are resistant to the hydrolysis. That's why they are called as a non-hydrolyzable tannin or condensed tannin, non-hydrolyzable tannin or condensed tannins. They are nothing but they are related with the flavonoid pigment. They are related with the flavonoid pigment. They are the derivatives of this catechin or flavon 3 or flavon 3 for diol. They are the derivative of catechin or flavon 3 or flavon 3 for diol. They are upon the treatment with the acid or enzyme, they are polymerized and decomposed into a red colored substance known as a propapanes. Then you will try to hydrolyze this condensed tannin with the acid or enzyme. So they get polymerized into one red colored substance known as propapanes, which are water insoluble and gives brownish red color with the many plant. Brownish red color to the many plant. Whatever color of brown or red color is there in many plant, that is because of the condensed type of tannins. Dry distillation will give you nucleus of catechol. Catechol will be the product upon dry distillation. तो यहाँ पे hydrolyzable tannin का क्या है? Hydrolysis product Esic acid and gallic acid. Condensed tannin they are not hydrolyzable, but upon dry distillation you will get a catechol nucleus, and they will produce a brownish green color with the ferric chloride. Example of this condensed tannin, for example, green tea is there, cinchona, cinnamon bark, wild cherry bark, black catechu, pale catechu, coca, and cola. In these plants, they will have a condensed tannin. Next one is the pseudotannins. Another class is pseudotannins. So, what is pseudotannins? Will not obey gold-beater skin test. They will not give positive gold-beater skin test for a examples of such pseudotannins, for example, chlorogenic acid from coffee and Naxomica. Chlorogenic acid present in the coffee and Naxomica, it is a pseudotannin. Epicaponic acid present in the epicatechin present in the pseudotannins will not give gallbladder skin test positive. So there is one specific test known as a chlorogenic acid test for the pseudotannins. They extract these tannins are treated with the water and treat the extract with the ammonia solution followed by exposure to air. So slowly formation of green color indicate the chlorogenic acid test. Chlorogenic acid. This is the test for pseudotannins. So there are three kind of tannins again. I will try to. So hydrolyzable tannins, they are easily get hydrolyzed with the acid or enzyme. Upon dry distillation, you will get gallic acid and other. Okay. So gallic acid and ellagic acid are the hydrolysis product. Upon dry distillation, gallic acid will be get converted to one compound known as pyrogalal. Compound perichloride give blue color. Example is calotannine, pomnatcol, rhubarb, clove, chestnut, elagitonin from myrobalan and pomegranate bark. Then condensed tannins, they are not hydrolyzable, they will resist the hydrolysis. So for example, phlobatannins, proanthocyanidins, these are the example. They are much resistant to hydrolysis, that's why they are known as non-hydrolyzable or condensed tannins. They are related to flavonoid because of the derivative, they are derivative of flavon like catechin or flavon thiol or flavon 3-diol, 3,4-diol. So they are the condensed tannins. So example याद रखो यहाँ पे phlobatannins and proanthocyanidins. इसके ऊपर क्वेश्चन आ चुका है. So proanthocyanidins are the type of condensed tannins. On dry distillation, these will give you catechol. They will upon dry distillation will catechol, which produces green color with the perichlorate. For example, green tea, cinchona, cinnamon bark, wild cherry bark, and black catechu, pale catechu, Coca-Cola. So all these will have condensed tannins. Now next one is the pseudo tannins. It will not obey Goldwater skin test. For example, chlorogenic acid from the coca, ipicaconic acid from coke. I can ask you chlorogenic acid or ipicaconic acid or catechins. They are the examples of the pseudotannins. Examples are the so in pseudotannin will have chlorogenic acid test. Next one is the first drug from this category, that is known as a myrobalan. First drug from the tannin category is myrobalan. Biological source for this myrobalan is nothing but the synonym for myrobalan, that is known as chebulic myrobalan. It is known as a herde or haridki. You might have seen this herde or haridki in condiments. Biological and geographical source. It consists of a dried ripe fruits of Terminalia chibola. Terminalia chibola family Combretaceae. Terminalia chibola family found in the some sub-Himalayan tracts of Ravi, then West Bengal. It is also found in West Bengal, Assam, and some deciduous forest of India, especially in Madhya Pradesh and Maharashtra. It goes up to altitude of 1,800 meters. Fruits are collected from wild grown plant in the forest. फॉरेस्ट में जैसे ऑटोमेटिकली जो सीडिंग होता है उसमें से जो प्लांट रेज होता है उससे ही कल्टीवेशन तो कांस्टेंट आर कलेक्टेड मद्रास एरिया are very rich in the tannin. It consists of hydrolyzable tannins. Chebulic acid, then D-galoyl glucose are the hydrolysis product. Chebulinic, elagic, and gallic acid are the other constants. During the maturation of tannin, during the maturation of plant decreases, whereas acidity. So what are the uses of this myrobalan? It is astringent, laxative, and stomachic. Tonsilitive property of this is due to the anthracene in pericarp of the fruit. Fruit make anthracene derivative, that's why it is a laxative. Otherwise, tannins are are used in the diarrhea. It is an ingredient of Triphala Ayurvedic profession. Triphala Churna, that is ingredient of Triphala. Commercially used in the dyeing and tanning industry. Also used in the treatment of water for the locomotives. Then it is used in the treatment of piles and external ulcer. Piles and external ulcer. So laxative है, astringent है, stomachic है, tonic है. Triphala Churna का ingredient है for the water locomotive. Commercially used for the dyeing and tanning industry. It is also used in the treatment of piles and external ulcers. Next one is the Behera. First was a Harda, or next is a Behera. Behera is also known as a Belleric myrobalan. Belleric myrobalan is a Harda. Behera is a Belleric myrobalan. It is known as a Behera, or in some language it is also known as a Badi Ilachi. Or it is also known as a Badi Ilachi. Biological source, dried ripe seeds of fruit known as Terminalia bellerica. Again, comparatively. Found in the Madhya Pradesh, Uttar Pradesh, Punjab, Maharashtra, and Sri Lanka. Sri Lanka. Cultivation is not done on a commercial scale. But is carried out by sowing the seeds to 30 days of germination of seed. They are transplanted. Chemical constant क्या है इसमें 20 to 30% gallic acid, elagic acid, tannins, then bilamine, ethyl gallate, and some non-edible oil. Is the use of this plant? It is astringent. It is used in treatment of dyspepsia and diarrhea. Dyspepsia means absence of gastric juice or disturbance in the digestion. It is also used in treatment of diarrhea. It is a purgative property of half-ripe fruits is due to the presence of oil. तो अगर ये पूरा पका हुआ नहीं लिया आपने फ्रूट एस्ट्रिजेंसी की वजह आपको पर्गेशन देगा टू So fixed by in the unripe, that is unripe fruit or half-ripe fruit. We'll see the next drug is Arjuna bark, Arjuna bark or Arjuna. So it's a bark obtained from this plant. So synonym क्या है Arjuna bark? Biological source dried steam bark obtained from Terminalia Arjuna family again Combretaceae. Terminalia cibola that is a Chebolic myrobalan. Terminalia bellerica that is a Belleric myrobalan. Terminalia Arjuna it is known as a it alone it belongs to family Combretaceae. All belongs to family Combretaceae. It is common in the Indian Peninsula region and very common in the Chota Nagpur region. Terminalia Arjuna. Division collection is not important. Plant is required for plant required real loamy soil 70 to 190 centimeter rainfall. Seeds take 21 days for germination. Chemical constituents for 15% tannins which are hydrolyzable. Hydrolyzable tannins 15%. Phenol, saponin, arginolic acid, and arginogenin, beta-acetostyrol, and ellagic acid. Then it also consists of one flavonoid known as arginidine, arginodone, and arginone. In chemical test, important. Extract of argina will show pinkish fluorescence under UV. Then uses are important. So it is a cardiotonic. Cardiotonic like your rest of the cardiac glycoside. So cardiotonic, but not so potent. Digitalis is very important. Styptic febrile. It is used in the dysentery, diarrhea. Also diuretic and tonic properties. Diuretic activity of Arjuna is due to one acid known as arginolenic acid. Arginolenic acid is responsible for the diuretic property of this plant. Essentially used in the tanneries and also used in the timber. Adulterants is adulterated with the dried bark of Terminalia tomatosa, used as adulterant and can be distinguished by person's taste. So it will give your original drug will give you pinkish fluorescence. It will not. You can see this is the plant of your Arjuna, Terminalia Arjuna. It is Arjuna bark. You see the next plant known as a Indian gooseberry or Amla. Indian gooseberry or Amla. So synonym for Amla, Indian gooseberry, Imblica. What is it? It is a dried ripe fruit of plant Imblica officinalis, mainly Euphorbiaceae. So not dried fresh fruits of Imblica officinalis, Euphorbiaceae. It is also found in the deciduous forest of India, Sri Lanka, and Myanmar. Cultivation collection. So it is grown by seed germination, but also can be propagated by budding and cutting. So we plant part of the stems ended. So it can be raised by stems cutting and budding, and does not tolerate frost or drought. So this plant will not tolerate इसको अगर पानी नहीं देंगे तो ये मर जाएगा. Not tolerate frost or drought. So varieties of this amla like Banarasi variety, Kanchan and Anpalu and 6 in 1 years will give you better yield. And require red loamy coarse gravelly soil. Seeds are sown in the at a distance of 4.5 to 4.5. For irrigation is required. Drip irrigation is most suitable. Fertilizers in a range of 750 and 100 plants via 1 kg super phosphate over there. So it is a internal cropping is possible. Is if you cultivating this amla plant, in between amla plant you can take another crop like tomato, guar, then sunflower, groundnut. These can be taken as an internal crop in the rows of this amla. Then hormones like gibberellic acid and hormones like gibberellic acid or phenolphthalein in a range of 30 to 50 ppm. They are useful to increase the yield. Now chemical constituents 600 to 750 milligram of vitamin C per 100 gram of fresh pulp. So if you obtain a fresh pulp of 100 gram of amla fruit, then you will get around 600 to 750 milligram of vitamin C. That is ascorbic acid. So fruit is a rich source of your phosphorus, iron, calcium. Fruit consists of a rich source for iron, phosphorus, calcium. So has a 5%. So that's why this amla is also useful in these tonics. So it is also ingredient of tripalachuri. Taste. Episode alcoholic extract of this amla will give blue color with the perchloride, blue color with the perchloride, aqueous extract plus gelatin and sodium chloride, milky white color. Milky white is due to the precipitation of gelatin. In the precipitation of gelatin. Aqueous extract plus lead acid. Largely used in the Indian medicine, it is used as a acidic, diuretic, refrigerant, and laxative. It is used in the diarrhea, dysentery, jaundice, dyspepsia, and amla oil is used in case of shampoos and hair dyes. It is an ingredient of popular Ayurvedic formulation. It has an antioxidant property also. Hair tonic. It is a hair tonic. It has the richest source of vitamin. The richest source of vitamin. Next drug is the Ashoka. Ashoka synonym is Ashoka bark. As source nothing but the dried steam bark of plant. Saraga indica family Leguminaceae. South Asia, Malaysia, Indonesia. It is the most sacred tree of Hindus. में ये proposed है Ashoka tree. It is frequently grown as an ornamental plant, not cultivated on. Also consists of 6% tannin, xylene, then testosterone, saponin, and organic calcium and iron. Testosterone is androgenic in nature. So androgen level आपका बढ़ जाएगा, मतलब testosterone वगैरह. So male hormones बढ़ जाएगा. Glucoper lagradinin is glucosinidine glycoside isolate from copper. Glucopyranidine, glucopyranidine, glycoside often formed. T of drug is due to the presence of steroidal component. Contain powerful oxytocic principle. Oxytocic है ये. Androgenic also and oxytocic. Oxytocic means it will increase the uterine contraction. Yes, क्या है? Uterine tonic, sedative, stimulant effect on endometrium, ovarian tissue, menorrhagia. Should not be confused with the garden tea. जो garden में जो अशोका plant रहता है, that is different. अशोका plant is तो garden में जो अशोका plant रहता है, that is a Polyalthea longifolia, which bears a white flower. Next drug is a black catechu. It is known as a katha, kach, or kadir, or catechu. It is a dried aqueous extract from the hardwood of plant Acacia catechu. There are two methods for manufacturing. Traditional method is there, and modern method is there. So in traditional method, so it is nothing but the hardwood. It is the extract of hardwood. This extract from hardwood of Acacia catechu leguminous. So in the traditional method, what they used to do, separate the hardwood and separate the hardwood from the earthen vessel. While that in an earthen vessel, till soluble portion is extracted, it is allowed to become semi-solid by natural cooling. Cooling, the less soluble fraction will separate out. The less soluble fraction is known as a semi-solid mass is known as a kach. It is transferred into rectangular pits at the bottom of which there is a clay and sand over there for several days. Each part is completely adsorbed by clay. In case of modern method, we have to use different extractor. Hardwood is cut into chips mechanically and put into steam extract. Extract is concentrated under the vacuum and cooled in the centrifuge to isolate the cakes of the liquor behind centrifugation. Left behind the after centrifugation is nothing but the kach. Next is what are the chemical constants present in this plant. So chemically consists of a 10% Aka-catechin. Aka-catechin is a diastereomer of 5,7 tetrahydroflavin 3. Aka-catechin is also known as acacia catechin. Catechin will undergo oxidation to give Catechu tannic acid. Another content like quercetin and quercetin. It does not contain fluoride. It is a non-fluorescent. So this acacia catechu will not have chlorophyll. It is obtained from a hardwood of the plant. So it is not obtained from leaves and stems. That's why it will not have chlorophylls. It is non-fluorescent. Another variety is known as a Amber Catechu. That is a fluorescence in nature. Chemical test: pink or red color aniline hydrochloric acid because of presence of catechin. Aniline hydrochloric acid because of presence of catechin. Then match stick test positive देता है ये. Aqueous extract plus lime water. Extract of drug plus lime water gives which turns into red precipitate because of some. Again tannin solution plus ferric chloride and ammonium sulfate. Sorry, tannic acid plus ferric ammonium sulfate will give you green color. Addition of sodium hydroxide, the green color turns into this astringent is used astringent for external boils. Head वगैरह आ गए ना तो इसमें यूज करते हैं. Then skin eruptions, ulcers, also used in the cough and diarrhea, cooling and digestive properties. So it is used for dyeing and coloring softening, then reducing the viscosity of drilling mud. ड्रिलिंग मड मतलब जो हार्डवेयर होते हैं उसमें जो ड्रिल करते समय एक ये डालते हैं उसमें लिक्विड, दैट इज नोन एज ड्रिलिंग मड, टू रिड्यूस द विस्कोसिटी ऑफ ड्रिलिंग मड. Manufacturing of ion exchange resin. So these are the uses. Remember, for externally it is used for treatment of boils. बेड वगैरह आला आपला तर वो यूज करतो. So you can see here it is a acacia catechu, plant of acacia catechu. Then it is a wood of acacia catechu, and it is nothing but the hardwood. Whatever the red portion is there, that is known as the hardwood, and it is obtained from it is aqueous extract of this hardwood. इसका extract है ये acacia catechu. It will look like this. आप अगर पान खाते हो तो उसमें भी यूज करते हैं acacia catechu. This one is next one is a pale catechu. This one is a pale catechu. We'll see about this plant, pale catechu. Next one is a pale catechu. So it is known as a Gambier catechu. It is known as a Gambier or catechu, pale catechu. So it is obtained from aqueous extract of leaves. Now it is aqueous extract of leaves and young shoot of the Unicaria Gambier family Rubiaceae. So due to it is extract of leaves and young shoot of plant, it will have chlorophyll in the structure. Okay, it will have chlorophyll, but your acacia catechu will not have chlorophyll. Now it is propagated cultivation collection. It is found in the Southeast Asian region, Malaysia, cultivated in Singapore and Indonesia. Cultivation collection and propagation is done by seeds in the damp soil. Nursery beds are raised, and after 9th month of that, the seedlings are transplanted in open field. Seedlings so seedlings are transplanted into field. First harvesting is done in the when plant is up to 2 meter height. The leaves and young shoots are collected and boiled in the pot called as a cauldron. Cauldron नाम का एक pot होता है उसमें. Then made of wood and iron at the bottom. 3 hours decoction is obtained and concentrated till it becomes pasty mass with yellowish green color. Chemical constant है इसमें catechin 7 to 33%, catechu tannic acid and Gambier rosin. Chemical test. The major difference is the chemical test. They can ask question on this chemical test. So again it will give positive mastic test. Then vanillin hydrochloric acid test will be positive. But the major difference between acacia catechu and your pale catechu is the Gambier rosin test. Okay, Gambier rosin test. This was question in GPAT 2021. What is the difference between pale catechu? The difference between pale catechu and pale catechu and acacia catechu can be differentiated by this Gambier rosin test. This was question in GPAT 2021. Okay, the drug is extracted with the alcohol and add sodium hydroxide and few drop of light petroleum. Then shake the mixture, keep aside for some time. Green fluorescence is observed in the light petroleum layer, which is a specific test to distinguish between black catechu and pale catechu. Black catechu means our acacia catechu. This is a pale catechu. This is an important test. Here इसके बारे में GPAT 2021 में question था last year. Gambier fluorescence test. Now chlorophyll test. As I have told you, this plant is nothing but the extract of your leaves and young shoots. That's why they have chlorophyll. So drug plus chloroform mixture. Okay, then drug plus chloroform. Warm the mixture. Filter in porcelain dish. Evaporate to dryness. Due to presence of chlorophyll, it will show green color. Now, what is the use of this pale catechu? It is used as astringent in treatment of diarrhea. treatment of diarrhea, local, astringent. It is also used in the lozenges. These are the uses. Just remember the difference between Acacia catechu and this Gambier catechu. Acacia catechu is known as a black catechu, and this Gambier catechu is known as a pale catechu. Okay. The last drug of today's session, that is a pterocarpus. Pterocarpus. So it is again a tannin containing drug, pterocarpus. It is also known as the Indian kino. The synonym for this plant is a kino or Indian kino. A biological source, it is a dried juice obtained from Pterocarpus marsupium. Dried juice obtained from Pterocarpus marsupium family Leguminaceae. It is found in the Gujarat, Madhya Pradesh, Uttar Pradesh, Bihar, Odisha. It is also found in the Karnal and Kerala, West Bengal, Assam. So what is the chemical constant? So 70 to 80% ketotanic acid. It is a leukocidal tannin. आपका जो ल्यूकोसाइड है ल्यूकोसाइड्स उसको किल कर देगा ल्यूकोसाइडल टैनिन आपका जो ब्लड में ल्यूकोसाइड्स होता है उसको किल करेगा so 70 to 80% ketotanic acid which is a leukocidal tannin and there is one compound known as quinored which is anhydride of quinoine is also there. So it is anhydride of quinoine. Quinored is anhydride of quinoine and more dark than ketotanic acid. Then also consists of K pyrocatechin. Identification test is not important. Leave it. Uses. So quinoa is a powerful astringent in treatment of diarrhea, dysentery, and positive hemorrhage. It is also used in the toothache. It is used in the diabetes. It is used in the diabetes and also used in the dyeing and tanning industry and printing industry. Now this pterocarpus has a and it is a hypoglycemic action, so it will decrease your blood sugar level, which is this property is used in the diabetes. So इस प्लांट के कप्स किए जाते हैं कप्स टी कप cups made up of wood of quinoa is available for treatment of diabetes mellitus with the खादी and ग्रामोद्योग कमीशन खादी ग्रामोद्योग कमीशन है उसमें ये किनो के कप्स मिलते हैं वो कप्स फॉर ट्रीटमेंट ऑफ डायबिटीज सो इंडियन किनो plant will look like this. So this was all about your tannins. So tomorrow we'll start our drug acting on CNS. Drug acting on CNS. Initially, in morning session, we'll see the nervous system, introduction to nervous system and all that. And after that, we'll see drug acting on CNS. So initially, we'll try to finish that general anesthetic chapter, then sedative hypnotic chapter. Okay, so tomorrow we'll take three chapters. One chapter from anatomy and two chapters from your pharmacology. Is that clear? Hello? समजलं का आज काय जास्त काय हे महत्वाचं नाहीये